First of all, thank you to all participants and the speakers for this webinar. This webinar is being organized by RI Hub's project, of course, counting with support of many other people from Euro-BioImaging, Latin America BioImaging, Instruct, and SEBEN. Please note that we'll be recording the speakers session, and then when we move to Q&A, it will not be recorded. So it's my pleasure to introduce the first speaker, Greg Kitten. So Greg graduated from Texas Tech University in biology, and he worked there as an EM technician and imaging scientist for 4 years before pursuing graduate studies in anatomy and cell biology. After a postdoc at NIH and roles at the Swiss Cancer Institute and the University of Iowa, he became a professor at Federal University of Minas Gerais in Brazil in 1997. So at UFMG, he directed the Light Microscopy Course Center and co-founded the Center of Microscopy, where he is currently the vice director. This national facility now serves academic and industrial users through all Latin America. Greg is involved with Global Bioimaging and Latin America BioImaging, and he chairs the Cryo-EM Project Evaluation Committee at LN-CNPEM, and he's also co-PI of 2 Chang Zuckerberg Initiative projects focusing on training imaging scientists. So Greg, thank you very much for joining us, and the stage is yours.
Thank you very much. Let me try to share here again. See if it looks okay up. Let me go back out. I think I'm going to have to stop share. Is it correct now?
Yes.
It's again the presenter view, so you have to do the display swap again.
Do it again. Yeah. Went back to the.
It's under the display settings at the top left.
Yeah, it's not showing for me this time.
Add one to the right, Greg. Display settings right next to it.
Yes.
There we go. Okay.
Yeah, there we go.
Okay, so I'm going to talk to you about the Center of Microscopy at UFMG in Brazil. It's a central national center, multi-user, multidisciplinary. And I have to apologize. I'm going to have some slides in Portuguese. Ran out of time to change, but they'll be obvious of what I'm talking about. So let me go to the next. So the Center of Microscopy, it has the goals of being not just a microscopy. I always have to explain that here. So we do a lot of microanalysis. So that will be obvious here in a few minutes, but a lot of energy dispersive spectroscopy and other electron microscopy. So it's academic and industrial communities are served. That's what I'll talk about mainly today. I want to tell you how it's been a huge advantage for us to have this connection with industry. Multi-user, multidisciplinary, big core center, I'll describe that. And we focus on both life and material sciences. So we provide services, we help people with research and development, we have our own innovation, and we do lots and lots of human resource training and career development. So the question before I really start, I have a few questions that I want you to keep in mind as I'm going through the talk is, do you really want to do services for companies in your core center? I think that's a question. It's a lot of work. I mean, it's an immense amount of work doing consulting with the people when they first come in, explaining, but usually they don't know what they need. Contracts, accounting, constant deadlines, pressure because you can't stop with sample analysis when you're working for these companies. You always have to be productive. And so the question is it worth all the work? A few other things I want to go quickly through these is, do you already do service for your companies in your core center? And if yes, how often do you do this service? Is it continuous or just occasionally? If continuous, do you have enough people trained to provide uninterrupted service when a technician is on vacation or sick? So that we've gone through that where we've had problems. So you have to think about this very much in beforehand. Are you confident you can reproducibly provide results for hundreds of samples week after week after week? So you have to know that you have the expertise and that your systems can give you reproducible results. Are there target companies? How big do you want to grow? If you go to many, you'll see ours is more on material side, but target companies now for me are pharmaceutical companies. So I'm trying to expand this into pharmaceutical, but you have to always be careful that you don't overextend your capabilities. Here's a good question for many of you that are in academic situations. So how much service should you provide for companies? At the moment, we provide 47% of our time is for companies. What happens if we go over 50%? So we have a big discussion about this. How much time should you allow companies to use of what was purchased with university or governmental funds? Something important also is, are there any restrictions on receiving and spending the funds that you've received? Do those payments come into your account or do they go somewhere else? This was a huge problem. When money goes into the accounts at the university that you don't have control of, you'll never see that money. So you have to have lots of discussions and be sure that you can use that money for what. And usually I'll tell you this a number of times that people told us, no, you can't do that. And when you press them, why can't you do that? Because we've never done it that way. So that you can do it. So you have to keep pressing, and then usually now things for us are working extremely well. So, and this has come up several times recently. I'm a little surprised. Do startups get any sort of discount? So startups often have governmental funding. They have, sometimes they're within the university. So should they get a discount for us doing service for them? They're not really a company yet, but startups. And so we also have had on several occasions that we're helping companies actually do research and development and had a lot of input. So we're always sure now that we put this into contracts. If there's a lot of input from us of intellectual knowledge, we want to be considered in patent applications. I'm going to jump back a little bit because I have to explain what our center is. And you can see on the map here, there's a little arrow. I don't know if you're seeing my mouse here, but circling the state of Minas Gerais. For those of you in Europe and the US, Minas Gerais is a little bit smaller than France and a little bit, no, I'm sorry, it's a little bit larger than France and a little bit smaller than Texas. So it gives you an idea of the size of the state. I'll come back to this in a minute, a fairly large state with, and I'll just mention a lot of mining industry. So here in Belo Horizonte, which is a town of about 6 million people in the metropolitan area. So this is the campus, which is 800 hectares of land and a big football stadium famous. But here in the campus, we have the main campus, and then hidden off in a very stable region of the campus is our center. You see with the arrow, yellow arrow in the image, and then a frontal view of this building here. So this building was conceived when a lot of microscope systems in engineering, biology, geosciences were having problems back in the early 2000s. So we decided to build a multi-user, multidisciplinary where we could put microscopes that were working into this building and then also combine the technicians and do good training. And so we called people from engineering. We traveled all over the world to make a plan for this building. And it's very well known here in Latin America. It's got a very low level of vibration, controlled magnetic field, thermally stable, and uninterrupted energy system. So it's a very nice building that's been used as a model for several places. The next slide will show the layout of the two wings that we have here. So we have a preparatory wing, which has 7 laboratories for preparation of samples. And so again, that's both for biological and for material science. Some things are similar, other things are quite different. A couple of laboratories or rooms that we have technicians staying. And then we have the big labs here. So we have 13 laboratories for microscopes. They will be all full, another microscope arriving soon. So then we'll be out of space here for microscopes. Back here, we have the generators and things that we have battery backups. And we also have a small building on the side that we have a classroom for teaching, which is getting a lot of use in the last 2 or 3 years. So we have lots and lots of courses and training. Down at the bottom, you can see several. These were 2 of the new microscopes when we originally opened and got the project funded. And then other microscopes. This is a scanning tunneling microscope, which came from Hewlett-Packard. People that were working there that had contacts, they said, you can have it if you bring it to Brazil. So we packed it up and brought it to Brazil. So we have lots and lots of equipment. I'll show you that, I think, here. So at the moment, we have 2 transmission scopes, 1 other new one arriving, several scanning electron microscopes, electron probes, scanning tunneling, other things on the list here. Only 2 optical microscopes. There's another center at our university that has all the fluorescence and super resolution, but we keep 2 just mainly for people to check and do a little bit of CLAM experiments. We are one of the few places, Rodrigo Portugal and also in Rio de Janeiro, they also have capacity. At the very moment that I'm sitting here, we have a cryo workshop that's going on at the moment. So we have 16 people participating in a training session for cryo-electron microscopy, low level for what we do, but we do freeze substitution and all of these things. So there's just the cryo equipment showing quickly that we have that capability. Here's the new microscope that's coming, will be arriving in 2025, but also capable, will give us intermediate cryo resolution. We want to just mention, because now we just recently, we're implementing serial section array, volume EM. We're not doing, we do have, don't have cryo-fib, but we have fib-sem, so we can do fib-sem tomography, but we're going to focus on serial section array tomography. So we have the system, it's already been installed. We're training now, still doing pilot tests with it. So where you make the 3D, and we're hoping to make these sorts of libraries so that when we do this, we do it with both neuroscience, where it's used heavily, but also with neglected tropical diseases, so that we can make libraries of samples collected from Zika, dengue, other materials that we can provide to people around the world for further analysis. I'm going to jump this just to mention, would show you this, but just because of time, I jumped this video. But we do have a close connection with Jeff Lichtman's lab at Harvard. So one of our member of our council of directors always spends January and February in his lab working. So we do have the expertise to do this 3D volume EM. I also want to mention that we just received funding for a TOF SIMS. And so this will be a huge, huge plus. We'll arrive maybe at the end of 2025. We're still trying to decide which unit we want. But this will be a huge, huge addition for biology, but even more so for material science. Let me go to the next slide here. So we have just to focus here on the different types of mass spec, but SIM, secondary ion mass spec, has a very high level of resolution. And so with this, we can detect all sorts of lots of publications starting to show up in the literature using TOF SIMS. We've set up a good network already with 2 groups, one in London at National Physical Lab, and the other in Maastricht in Holland. So we're trying to set up a network, and Argentina here, and 2 places here in Brazil. So we're setting up a TOF SIMS network. This is, sorry, I have this in English somewhere. I couldn't find it when I mounted this and didn't have time to change it. But essentially what I'm telling you here is the council, Conselho de Diretores Council, Directors Council. And this is the coordination is a technical group that we have. So every microscope has somebody that knows it inside out. So we have specialists. And so when a new user comes in, whether it's from a company or whether it's somebody that wants to do something special and they have questions, we usually send them off to one of these professors to do if we don't know the answer, we send them to one of these professors. They discuss what's the best way to prepare their sample. So it's been a huge plus for us to have a group of volunteers that come in, and we send them off to somebody that knows atomic force microscopy of biological materials. So we have a person very good. They go talk and then set up, design exactly how they want to do the experiments. So that's one group. And then we have our technicians. And so this is hugely important for us because, as you'll see here, we have PhDs. So we have 12 PhDs, actually 13 now, and 11 master's student. I'm sorry, yeah, 11 people with master's student. Now one just got hers. So the numbers constantly change here, but usually we have 34, 35 people that are highly trained in the center. I'll come back to this in a few minutes, but with money from the university, we only pay this first line. So what are the permanent positions from the university, the 14 people. All the other people, we have to find funding from agencies, so the Brazilian funding agencies, or we have 2 of these people are on CZI grants, but we have to look for money to pay 20 people essentially on this block. So that will be important when I come in front a little bit more. We also do something, I just wanted to show this because when you talk about companies, you always have to be sure that you don't stop when somebody goes on vacation and these things. So we have level 1, level 2, and level 3. So level 1 is the people that they know the microscope system in and out. They've worked on it for years. They're the leaders on that microscope. But then we have other people that are still training, or they need to go back through and do a little bit more study. So every one of our microscopes and every one of our methods, we have at least 3 people in level 1, 2. Always at least 1 in 1 and usually 1 in 2 and level 3, so people training. Many of the systems have 2 or 3 in level 1. So not shown here in another graph, but it's always, we have to constantly keep pushing people because we want to not have just stay with one method. We want to form, that's our human resource formation. We want imaging scientists that are capable of using several systems and the preparation of samples. When you have that sort of everything I've shown you so far, we have to have a very good hierarchy of everybody knows who's responsible for what, good communication between all the people. So we have a supervisor of microscopy, and they have the coordination team, the council of directors here, and then we have all the different microscopes. So each one of these has a person that's responsible. So when we have meetings, it's like the people that are here with the scanning electron microscope. They don't come straight here to the director. They talk to people through the hierarchy, the system, and then the problem comes. So it's a very efficient system at this point. With all of these people and all of the things that were going on, I'm not going to talk about our application for certification. We went through all of that. We have all the documentation, and I do think here, so we prepared to go for certification, but then here in Brazil, it's very expensive, and we couldn't see the cost benefit for us at the moment. So we did not go for certification. ISO certification, we waited. Maybe someday, but so there are 8,769 documents, and there are 1,000, almost 1,300 files. So we're very oriented to keeping track. If you have 20, 30,000 samples coming to your center like we do every year, you can't lose control of billing and samples and all of the things that go through. So we have a very good system to follow all of that. Our webpage, most of it, you can click on the little things here and it'll go for English or Portuguese. I think now it actually also has Spanish on the little icons here. So you can go in and you can see everything about the instruments. So we have a fairly good web page. Any of you that have web pages, or they're always under construction. We have table of cost, as I've indicated here. And so another point I want to just show you here is, so the way that we do this, so at the university, you pay one value. Other universities and other institutes of research, they pay twice the value. And then companies in this, they pay a varied value. In this case, in this particular thing, it's 330, so 3 times the value. Other things, if you look down here for using different microscopes, there are different ratios depending on the complexity of the analysis. A good example here for one of the scanning electron microscopes is 150 PIs internal, 250 external, and 850 for companies. And so these vary a lot too, depending on the samples. So the cost for companies usually varies anywhere between 4 and 10 times the value that we pay within the university. So we make these calculations very carefully, discussing also with what they pay in the market if companies had to send it elsewhere. Our prices are usually much below what they would do if they had to send it to somewhere else, to Australia or US or somewhere else. At the moment, we have, this is a little bit behind, but we have roughly 1,200 actually registered users and about 1,400 projects total. If you look at these numbers, so you see in the kind of orange color here is UFMG, and so it steadily grew since when we started keeping really contact or control the numbers. And so we've grown progressively over the years, and I think this is something important. You don't want to grow too quickly. I think for us, it's a very controlled growth. We only do this when we feel we have the expertise to do high quality work. The point here I have this circled is because in 2022, other institutions passed our own university as users of our system. That if I can show you here, so this is a map of Brazil, and all of the states colored in blue have at least two institutions are using our center. Others have more, 3 or 4, or in our case, there are 36 institutions in our state that use our center of microscopy. I just checked. This is the numbers of projects that are actually active per year. And at the moment for 2024, I don't have the number for 2023, but at the moment we have 204 active projects up until this moment. So probably will be around 205, 210 projects active this year. When you look at what does all of that do, all of that work produce? So we have to, of course, always make the reports on production. And so in 2020, so pandemic, we had 191 publications, it's probably better seen on the graph down here. But we also have, if you look up here on the top, we're in the levels, we're recalculating these numbers. There was some discrepancy, but we're roughly in the 60 masters and PhDs per year that are produced using the Center of Microscopy. So we have good numbers and lots of users, lots and lots of production. Some of those are usually high quality, so a lot of publications in Nature and Science, getting also awards, some best pictures, and attention of discovering new viruses with no recognizable genes using, well, not that obviously we don't sequence, but we did all the images for that paper. Other things we do, we do a lot of liposome. We don't have high resolution cryo-EM, but we have lots of academic people doing liposomes and nanocarriers for drugs, not only for companies, but also for academics, but also lots and lots of work for companies. These are just 2 other from material science, front page of Nature and other things. So we have a very good high publication rate and a very good high productivity on these. We do lots. Usually, I think this year we'll probably pass somewhere between 700 and 800 biopsies. This is a point that I'll make now, but we'll come back to later. So in this case, we were doing all of the preparation and doing all of the microscopy. In this case, they finally decided, we trained the people from this company to do this themselves. So now they come and are autonomous on our microscopes, and they purchased equipment. So they purchased microtomes and materials. So they do their own embedding, they do their own sectioning, and then they come here and only use the microscopes, usually at night. So that our systems are used very heavily by this company that does this diagnostic biopsies here in Brazil. Now, going to users, let me see, right? Oh, no, okay. So the users from companies, so we have 121 companies at the moment, maybe a little bit more, but in August it's 121. And so a lot of them are from different areas, but kind of indicated here this is from mining companies, but then we also have a lot of advanced materials, sometimes linked to these. Mechanica. Some of these are all linked to the mining industry with iron and different ores, aluminum that they're using, automotive also, but other material science. The life sciences is in this range here, so not obviously far below what we have on the other side, but we're trying to expand this, and I think we're actually a little bit above these numbers here, but I'll show you a list in just a minute. So we get money from this service from all of these companies, but we also have, obviously, we have several grants from state institutions. We have two different Chang Zuckerberg grants, and we have money from the federal institutions. So our funding comes from various sources. If you look at a map of Brazil, this gives you an idea. This is if you can read it there, it's under mine. These are from mining and metals that are present. So iron, precious metals, other base metals, so a distribution across Brazil. And we're we're more or less where the number 18 is. And so we have a lot of iron in this area and other aluminum, other materials in the vicinity. But we also have lots of lithium and niobium and other metals. Other niobium is very rare. So we also have projects with the companies that do niobium. So let me just showing you a little bit more of that same kind of list. So here are some of the numbers for the specifically out of that graph, but the life sciences here, you see we have environmental, pharmacy, pharmaceuticals and biomaterials, equipment, biomedical equipment, diagnostic, for health diagnostic and food analysis. So here are the list of those companies that we have, then 17 or 18 companies here.
Sorry, just to mention, we reached 25 minutes. So
okay, so time or okay, I'm going to shut here in just 1 second. So with these, so quickly, we have, I won't go through this, but we have our budgets, and usually what we bring in from these companies are on the value of 400,000. This year will probably be between $400,000 and $500,000 that we bring in from service for companies. And then, didn't save. So I had some questions there that I'm not sure why it didn't come up on this, but is it all worth it? For us, yes, it is. I think it's good for the companies. It helps us build the infrastructure here in Brazil so that companies have access to the best technologies. We have a lot of other connections with other systems that do building the ultrastructure for Brazil. But the point I would say in the long run for us, it's very useful to have these interactions, but it is a lot of work and you have to have a good team that communicates with each other. So these I'm going to jump over. I just want to talk about, this was with a Labby, not with Labby, but with the BioImaging net. So we just had a meeting a few weeks ago. So it's growing very much here in Brazil, and have different, I don't have time to talk about any of these, but we have a lot of outreach programs and then programs now, because of global bioimaging, because of Latin American bioimaging and Chan Zuckerberg, we've developed several collaborative projects across South America, but also with Germany, England, and other places. So I'll stop there and just tell you, if you're ever in Brazil, please contact me and we'll give you a tour of what we love for our Center of Microscopy. Thank you very much.
Okay, Greg, thank you very much. We'll now move to Julia. Julia Fernandez-Rodriguez is the head of the Center for Cellular Imaging and Integrated Microscopy Unit at SciLife Lab in Gothenburg. Also co-director of the Swedish National Microscopy Infrastructure. She served as a vice chair of Euro-BioImaging panel of nodes, and she's the president of the Core Technologies for Life Science Association. With over 22 years of experience in cell and molecular biology, she leads a relative multimodal imaging facility where she integrates advanced imaging technologies and analysis. Julia is also part of various national and international scientific advisory boards and review panels. So Julia, thank you very much for joining us, and the stage is yours.
Thank you very much. Let's share and go. I will go a bit more general in a way, and using my facility as an example. And as Rodrigo said, yes, I mean, I'm running a facility in Sweden. Now we are in the North Hemispherium and very north into Europe. This core facility, just going in a half. We were supposed talking about facility management, and I just bring up some of the points I think it might be important, and for all of us in general, not only for in case of my facility, but I just use the Center for Cellular Imaging as. Let's first focus on local and regional, if we are actually management. I just showed you what is our main organization. Our facility are part of the Gothenburg University. Gothenburg University have two main faculties. One is the Sahlgrenska Academy, that is the medical faculty, and the Faculty of Science. There is, and of course, we also interact a lot with Chalmers University. This is the other university that is just a kilometer away from the Gothenburg University. And this is a bit more technical university. But just where we are focused here is where we call the core facility organizations, that the head of these core facilities are under the dean directly. And then we have different type of core facilities, bioinformatics, cellular aging, proteomics, and genomics. This is just the heads of all the core facilities. It's also core of a main facility for the NMR in the faculty of science. And of course, there are a couple of facilities. Here is only the ones that they have for Science Life Lab. If I now focus on my facility, we are, as I say, a correlative, or just Rodrigo said, they are a correlative multimodal imaging, and you can see now why. We have different type of microscopes, fluorescent microscopes that go from conventional, confocal, multiphoton, super resolution, and light sheet. We also have electron microscopy focus on scanning and light microscopy. We do a lot of course, array tomography in the volume EM part, and of course we can also do tomography in our 10, and we are focused more into the life science. And we have some material science users, but the main of them are into the life science. And of course, because you have this multimodality, also we I collaborated with a professor that they do the multi-imaging mass spectrometry. And of course, when you have all these multimodality and correlative approaches that you want to jump in from one microscope to another, you definitely need a team that also can help us into the processing analysis. This is the team, this is the important people that they actually doing the job, and just showing you where we actually do for this managing. We are actually a centralized facility, and not talking about now local and regional. When I mean regional in this moment, I say the region of Gothenburg and at least the west coast of Sweden. And we are, of course, into the Gothenburg University, and the idea is reducing costs by pooling all this expensive equipment and staff, and try to avoid the duplication in the departments. Our mission, that means that is actually have expert service and consultation. We are a support unit for scientists that they can outsource complex projects that they require really specialized expertise. And I will emphasize a lot of the expertise. And this is actually going beyond the capabilities of individual research laboratories. We typically offer different range, as you see, from equipment usage, we training the people. We do also have service, and we do data processing analysis, and of course we can help with technical support and training, and we also do method development a bit more innovative in the facility. The managing is heading by a responsible that is the head, that in this case is me, that managing overseeing the operations, the budget, and the strategic leadership, and also the user engagement of the facility. And of course, for the highly training people that is actually behind and that helping me to maintain the equipment and provide the technical assistance. We do training and education. This is one of the main tasks and jobs that we have in the facility, to advance in the techniques and the technologies. We do a collaborative, we try to do a collaborative environment. We try to foster interdisciplinary collaboration, not just only in between the scientists and the people around us, but also with other facilities around. We are accessible. Obviously, we are an open access facility, and I have to say the first one that was in Sweden 22 years ago, we already were an open access facility. One of the majority of the facilities was actually more into departments and close into their institutions or departments. How the funding model, we actually have a combination where we have user fees and also the institutional funding that provides us with a sustainable financial structure. And the requirements to actually become a national or a University of Gothenburg facility, as we are now, is we have to have national importance. We have to have a broad user base, and we have to have at least 3 years budget plan, and there will be an external evaluation every 3 years, and we will have to show that we actually deliver it to our users and to the community. And obviously, to deliver the full potential, as I say, from these methods is from the methods that they will rely in the expertise and the facility staff. They are actually service oriented, of course. They understand the user needs, they try to push the technology, and they take pride in making the user successful. And I think this is the key from the core facilities or research infrastructures in general. If we don't have the right people, we always will enter in problems. That's what I say. If you're talking about facility management and we focus on local and regional, you need to have a fully effective and beneficial core facilities. You need to have the appropriate technology and human resources. This is important. Appropriate funding scheme and definitive institutional support, structural support to go ahead. It's actually very, very important, and we have been always very lucky that the faculty, that in our case is the medical faculty, the Sahlgrenska Academy, have been always very supporting, the same as the Gothenburg University. What happened now when we actually have a facility management for the national, international point of view? We are also part of 2 national research infrastructures, as Rodrigo have said. We have the national microscopy infrastructure that we get the money directly from research counseling, and the Science Life Lab, that is another life science big infrastructure in Sweden for the life science, and we get directly the money for them, and actually directly the money from the government of Sweden. And we are also an international research, part of an international research infrastructure as the Euro-BioImaging. And what do we do here? Well, here we support now cutting-edge research and innovation by offering access to unique equipment, tools, and of course services and the expertise. And they actually go beyond the capabilities of an individual institution. Now it's just not an individual lab like we have in the local or more regional, but now it's actually that it will be difficult for individual institutions have. It's just like we will have discussed now about cryo-EM or big instruments, light sheet microscopy, with all the methodology and all the analysis and processing that is included. Of course, the services here, they include as well equipment and technologies for super resolution, multimodal imaging, correlative multimodal, optical clearing, intravital systems, cryo-EM, etc. And of course, computing now, and we're talking about supercomputer, data infrastructure, or high-performance computer. Once we're talking about management of this kind of infrastructures where we are part of it, the managing that will become more as a consortium with the different institutions, and sometimes as well with governments, national governments, but also with international support. This is just an example of the people we have for the national microscopy infrastructure we have in Sweden. This is the 5 nodes that going through the whole Sweden from the west to the east to the north, and with the different technologies that we actually have in all this. And this is the people, because this is actually the ones making this happens. And as well, as I say, we are part of the Science Life Lab, and there is 2 main units in the platform that call the Cellular Molecular Imaging Platform, and we are focused here into the integrated microscopy technologies, in this case with the correlative array tomography as one of the pictures we have here. I will not show many pictures this time, or images. Thus, the mission at all this national infrastructure or research infrastructure is as well expertise, open access to state-of-the-art microscopy, enabling imaging across scale, and definitely to the expertise that we have. We also gain as well so much from that expertise. If now we come back to my facility example, we have, of course, we are in the Gothenburg University, do services, access, training, data processing, networking, innovation, and sharing expertise. We are part of the national infrastructures. We are actually part of a regional, Nordic regional. In this case, we call the Bridging Nordic Microscopy Infrastructure. Now region here means all the Nordic region that include countries like Norway, Sweden, Finland, Denmark, and Iceland. And of course, we are part of an international network as the Euro-BioImaging. All that, how do we deal with that? Well, we have some kind of hybrid financial model. We actually, being by open access, means that we have access from any kind of researches that come to us, that can be for industry, and as well, of course, from the academia, mainly of them from academia. If I just discuss about or show how the money that come into the facility, of course, we have the internal grants that come in from the structure, from the organization, from our institution, in this case, the Faculty of Medicine, the Sahlgrenska Academy. We get users fees, and we have external grants, and of course we get some money for teaching, and sometimes some extra monies if we are actually part of certain events or organizing events. If we now go to the budget distribution, obviously most of the money is going in salaries, and after depreciations of instruments, administration, premises, overhead costs, we really need to pay rents here. Some rents in Sweden are quite high, overheads and running costs. And all this, as you see, actually we have a quite big jump of external grants. This is positive and negative. Both, of course, they allow us to do more developing, because some of these grants are very related to innovation and developing. But at the same time, we very much depending on the period. That means when we have external funding, the use of this funding, it will be based into between 4 to 5 years, depends where the money come. Where when the internal funding is used only in the year base, and we have, of course, we have these 3 years ahead on the Gothenburg University, but at least we're using one by one and then the key difference, I will say, when we are like a core facility, local and regional here in Gothenburg, or we are more national and international, is the who is we typically serving. Obviously, when you have core facilities, it have to be more the institution or other organizations around, where research infrastructure is becoming a more large scale, and now we get more national, international. The visibility we have accessibility as well. Normally, it's accessible to institutions that host them, or regional here, where research are designed more accessible to a wide range of researchers. And of course, the funding management, I just showed you the fact that we get national funding allow us to actually be a research infrastructure, national and international research infrastructure. But of course, there is challenge and pathway to success. I want to finish with the success, not only with the challenges, but I just say one of the challenges, I think, when managing a facility is first the financial sustainability, which funding model will be the best for us, how if we depend on short-term grants, if this is a problem, how we get the long-term ones, and the sufficient funding. That this is one of the main things that we all go, that we have problems to actually make it operational, but there are more maintenance in the cost. I mean, if we don't have this maintenance of the cost, it will be very difficult to advance in equipment. That means that we will have limited funding, often leads to aging the infrastructure. It will be very difficult to change our instrumentation, that always is quite expensive. Interregional collaboration is really good, and we have, but of course it actually can also produce some challenges, because the funding priorities, administration and access policies are different, and sometimes that can hinder the collaboration across region. Even so, I think it's extremely important, and we have noticed that with our bridging Nordic infrastructure. And of course, sometimes there are access that they are not equal. I mean, sometimes some facilities are overbooked, other ones are underutilizing, and it's difficult. Scalability, if you want to scale in services, to me, you have to have a very clear way to actually, and a quality that will help you to scale without any problem that you will have against financial problems or insufficient funding. Of course, there are also technical challenges like maintenance and downtime. I mean, aging infrastructure, they limited the maintenance budget, and it is actually very difficult to deal with that. Upgrades and obsolescences. The technology now goes so rapidly advanced that the instruments become obsolete as soon as we get it. And it's actually, we need constant needs for upgrading, and this is a problem as well. Staff retention, I think this is the recruiting and retaining skilled technical staff in the core facility is actually difficult, and the competition we have in between academy and industry sometimes is difficult especially for the academia to deal with that. And I think this is one important point. Data management, obviously, I think is the fashion, the topic, and that is important in the whole for storing, sharing, analysis, and deal with all this data or big data that's coming. And if now go to my facility as a multimodal facility, we ended up not only with the data management in confocal or in this, but actually we have different modalities with different formats, different size, different data sets. Some of them are bigger, they can go to terabytes, and the size as well make it different if we want to process or analyze, because that's make it almost impossible to transfer or to cropping or to drift or to shading. And of course we have the topic of big data and how the post-processing data. The ideal situation is that we take data, we sort it, we arrange, we present visually, and we explain the story and get there. But the reality is this is us. This is what happened with multimodality. I will not enter in details, but definitely this is one of the big troubles, and that we as a head or the manager have to really deal and thinking about how we actually could deal with that. And as I say, building and operating and maintaining a state-of-the-art core facility is demanding.
Sorry to interrupt, just to let you know, we reached 25 minutes.
Okay, and just almost finishing. Pathway to success. First, join force and integrate with other facilities and alliance and infrastructure associations. Team working between core facilities and building alliance, as I say. Advocate for institutional support. I think this is important. Value on core facilities, operational funding and building resilience. Strengthen advocacy for public engagement. I think that's important as well, and have the user feedback and collaborative as well with policymaker fundraising agencies. Foster interregional and international collaboration for sure, and expanding partnership. And staff rotation, we really need to work on that, developing competitive careers for there, create collaborative environment where the staff feel valued and motivated, and offer competitive salaries for our people. Data management, as I say, I think is extremely important, and the visibility and outreach. This is recorded, you can see what is, I think, the success to that. And just to finalize, and as a take-home message, I think the important thing is prioritize goals, focus on addressing the most problems in the facility and align them, invest in people, I think prioritize staff, engage with the stakeholders, I think this is extremely important, users, founder, planning, decision makers, collaborated with success, plan sustainable, you really need to have focus on financial environments and have a really clear operational sustainability. Scale strategically, master data management, and celebrate success with everybody. And with that, thank you very much.